Welcome to our journey through the digestive system, a fascinating chapter in human anatomy and physiology. Understanding digestion is crucial for anyone interested in health, medicine, or biology. This system not only breaks down the food we eat but also absorbs nutrients and eliminates waste. By exploring its structure and function, you'll gain insights into how our bodies process meals and maintain balance. Whether you're a student, healthcare professional, or simply curious, this video will help you appreciate the complexity and importance of digestion. Let's begin by setting the stage for why this topic matters in everyday life and medical practice. Understanding the digestive process is essential for assessing patients with digestive disorders, such as acid reflux and heartburn. These conditions can significantly impact quality of life, and knowing how digestion works helps healthcare providers diagnose and treat them effectively. The digestive system's health is a window into overall well-being, influencing everything from nutrient absorption to immune function. By grasping these concepts, you'll be better equipped to recognize symptoms and understand the underlying causes of common gastrointestinal issues. Let's explore the key functions of the digestive system and see how they relate to everyday health. Digestive system has four main functions: taking in food, breaking it down into nutrient molecules, absorbing those molecules into the bloodstream, and ridding the body of indigestible remains. Each step is vital for maintaining energy, growth, and cellular repair. The alimentary canal, along with accessory organs like the liver and pancreas, work together seamlessly to accomplish these tasks. As we move forward, we'll examine each component in detail, starting with the gastrointestinal tract and its activities. This foundational knowledge will help you understand how digestion supports overall health. Gastrointestinal tract is a dynamic system, performing activities such as ingestion, propulsion, mechanical digestion, chemical digestion, absorption, and defecation. Peristalsis and segmentation are two key movements that help mix and move food along the tract. Peristalsis involves rhythmic contractions that push food forward, while segmentation mixes food and increases contact with digestive enzymes. These processes ensure that nutrients are efficiently extracted and waste is eliminated. Next, we'll look at the structure of the alimentary canal and its relationship to the peritoneum and peritoneal cavity. Peritoneum is a membrane that lines the abdominal cavity and covers most digestive organs. It provides support and reduces friction as organs move during digestion. The peritoneal cavity, filled with fluid, allows organs to glide smoothly. Mesenteries, folds of the peritoneum, anchor organs and provide pathways for blood vessels, nerves, and lymphatics. Understanding these structures is important for grasping how the digestive organs are organized and supported within the abdomen. Next, we'll explore the basic structure of the alimentary canal and its layers. Gastrointestinal tract has its own nervous system, known as the enteric nervous system or gut-brain. This system consists of enteric neurons that form two main nerve plexuses, the submucosal and myenteric plexuses. The submucosal plexus regulates glands and smooth muscle in the mucosa, while the myenteric plexus controls GI tract motility. These networks allow the digestive system to operate independently, coordinating movements and secretion. The enteric nervous system is essential for efficient digestion and responds to various stimuli within the gut. Enteric nervous system participates in both short and long reflex arcs. Short reflexes respond to stimuli within the GI tract, while long reflexes are triggered by stimuli inside or outside the gut, such as signals from the autonomic nervous system. The parasympathetic system enhances digestion, whereas the sympathetic system inhibits it. These reflexes help regulate digestive activity, ensuring that the system responds appropriately to different conditions. Understanding these neural controls is key to appreciating how digestion adapts to our body's needs. Three key concepts regulate GI activity: mechanical and chemical stimuli, effectors like smooth muscle and glands, and control by neurons and hormones. Receptors in the walls of GI organs respond to stretch, changes in osmolarity and pH, and the presence of substrates and end products. When stimulated, these receptors initiate reflexes that mix and move contents or inhibit digestive glands. Both intrinsic enteric and extrinsic autonomic neurons, along with hormones from the stomach and small intestine, coordinate these responses. Oral cavity or mouth is the entry point for food and the beginning of the digestive process. It contains structures like the teeth, tongue, and salivary glands, each playing a unique role in preparing food for swallowing. The mouth is responsible for mechanical breakdown through chewing and mixing food with saliva. This process forms a bolus, making it easier to swallow and begin chemical digestion. Let's take a closer look at the anatomy and functions of the tongue and salivary gland. Tongue is a versatile organ with several functions. It grips, repositions, and mixes food during chewing, forms the bolus, and initiates swallowing, speech, and taste. Intrinsic muscles change the tongue's shape, while extrinsic muscles alter its position. The tongue's flexibility and strength make it essential for efficient digestion and communication. Its surface contains taste buds that help us enjoy flavors and detect potentially harmful substances. Next, we'll examine the role of salivary glands in digestion. Salivary glands play a crucial role in digestion. They cleanse the mouth, dissolve food chemicals for taste, moisten food, and compact it into a bolus. Saliva also begins the breakdown of starch with the enzyme amylase. These glands ensure that food is properly prepared for swallowing and further digestion. There are three major pairs of salivary glands: parotid, submandibular, and sublingual, each contributing to the production and composition of saliva. Salivary glands are composed of two types of secretory cells: serous cells and mucus cells. Serous cells produce a watery secretion with enzymes, ions, and a bit of mucin, while mucus cells produce mucus. The parotid and submandibular glands contain mostly serous cells, whereas the sublingual gland consists mainly of mucus cells. This diversity allows saliva to perform multiple functions, from lubrication to enzymatic breakdown. Saliva is mostly water 97-99.5%, making it hypoosmotic and slightly acidic. It contains electrolytes, enzymes like salivary amylase and lingual lipase, proteins such as mucin, lysozyme, and Ig-A, and metabolic wastes like urea and uric acid. Protective components including lysozyme, Ig-A, defensins, and nitric oxide help defend against microorganisms. This complex mixture ensures that the mouth remains clean and ready for digestion. Salivation is controlled both continuously and in response to food. Minor glands keep the mouth moist at all times, while major salivary glands are activated by the parasympathetic nervous system when food is ingested. Chemoreceptors and mechanoreceptors in the mouth send signals to the brainstem, which stimulates salivary glands via cranial nerves VII and IX. This ensures that saliva is produced when needed for digestion. Teeth are essential for mechanical digestion, breaking food into smaller pieces to increase surface area for enzymes. Human dentition includes incisors, canines, premolars, and molars, each designed for specific functions like cutting, tearing, and grinding. Proper dental health is vital for efficient digestion and overall well-being. Next, we'll follow the path of food as it moves from the mouth to the pharynx and esophagus. Chemical digestion is initiated in the mouth, where enzymes like amylase start breaking down starches. This early stage of digestion is crucial for preparing food for further breakdown in the stomach and intestines. The combination of mechanical and chemical processes in the oral cavity sets the stage for efficient nutrient absorption later in the digestive tract. Pharynx acts as a passageway for food, fluids, and air. After food is chewed and mixed with saliva, it passes from the mouth into the oropharynx and then into the laryngopharynx. This transition is essential for directing food toward the esophagus while keeping the airway clear. The pharynx's structure and function ensure safe and efficient swallowing. Esophagus is a flat muscular tube that connects the pharynx to the stomach. It features the gastroesophageal cardiac sphincter, which keeps the orifice closed when food is not being swallowed. Mucus cells on both sides of the sphincter protect the esophagus from acid reflux. This structure is vital for transporting food safely to the stomach and preventing damage from stomach acids. Swallowing or deglutition is a complex process involving coordinated movements of the mouth, pharynx, and esophagus. It ensures that food is safely transported from the oral cavity to the stomach. The process includes voluntary and involuntary phases, with muscles and nerves working together to prevent choking and direct food properly. Understanding swallowing is important for recognizing and treating disorders that affect this function. Stomach is a temporary storage tank that starts the chemical breakdown of protein digestion. It converts the bolus of food into a paste-like chyme. The stomach can expand from about 50 ml when empty to up to 4 liters when full. Its structure and function are designed to efficiently process food and prepare it for absorption in the small intestine. Stomach contains several types of cells, each with a specific function. Mucous neck cells secrete thin acidic mucus. Parietal cells produce hydrochloric acid HCl and intrinsic factor. Chief cells release pepsinogen and lipases, and enteroendocrine cells secrete chemical messengers like serotonin, histamine, somatostatin, and gastrin. These cells work together to break down food, protect the stomach lining, and regulate digestive processes. Stomach is protected by a mucosal barrier, which consists of a thick layer of bicarbonate-rich mucus, tight junctions between epithelial cells, and rapid replacement of damaged cells by stem cell division. This barrier prevents the stomach's acidic environment from damaging its own tissues. When the barrier is breached, conditions like gastritis and ulcers can develop, highlighting the importance of this protective mechanism. Gastritis is inflammation caused by anything that breaches the stomach's mucosal barrier. Peptic or gastric ulcers are erosions in the stomach wall, which can lead to peritonitis and hemorrhage if they perforate. Most ulcers are caused by Helicobacter pylori bacteria, but they can also result from NSAIDs like aspirin. Understanding these conditions is vital for diagnosing and treating digestive disorders. Intrinsic factor produced by parietal cells in the stomach is essential for the absorption of vitamin B12 in the small intestine. Vitamin B12 is needed for red blood cell maturation. Without intrinsic factor, pernicious anemia can develop, requiring patients to receive vitamin B12 injections. This highlights the importance of stomach function beyond digestion, impacting overall health and blood formation. Stomach carries out several processes: breaking down food, serving as a holding area, delivering chyme to the small intestine, denaturing proteins with HCl, enzymatic digestion of proteins by pepsin, and absorbing lipid-soluble substances like alcohol and aspirin. The secretion of intrinsic factor for vitamin B12 absorption is the only stomach function essential to life. These processes ensure that nutrients are available for absorption and use by the body. Gastric mucosa secretes over 3 liters of gastric juice daily, regulated by neural and hormonal mechanisms. Vagus nerve stimulation increases secretion, while sympathetic stimulation decreases it. Gastrin, a hormone, stimulates HCl secretion in the stomach and triggers antagonist hormones in the small intestine. These controls ensure that gastric juice is produced when needed for digestion and inhibited when not required. Cephalic reflex phase of gastric secretion is triggered by the aroma, taste, sight, or thought of food. This conditioned reflex prepares the stomach for incoming food by stimulating gastric juice production. It demonstrates how the digestive system responds to external cues, ensuring readiness for efficient digestion. Gastric phase lasts 3-4 hours and provides 2/3 of gastric juice released. It is stimulated by stomach distension, which activates stretch receptors, and by chemical stimuli like partially digested proteins, caffeine, and low acidity. Enteroendocrine G cells secrete gastrin, which triggers HCl release from parietal cells and activates enzyme secretion. The buffering action of ingested proteins causes pH to rise, activating more gastrin secretion. Low pH inhibits gastrin secretion occurring between meals and during digestion as a negative feedback mechanism. The more protein consumed, the more HCl is secreted, causing a decline in pH, which then inhibits gastrin secretion. This feedback loop prevents excessive acid production and maintains balance in the stomach. Intestinal phase begins with a brief stimulatory component followed by inhibition. When partially digested food enters the small intestine, intestinal gastrin is briefly released, encouraging gastric glands to continue secretory activities. However, as the intestine fills, inhibitory stimuli override the stimulatory effect, protecting the intestine from being overwhelmed by too much chyme or acidity. Four main factors in the duodenum inhibit gastric secretions: distension due to entry of chyme, presence of acidic, fatty, or hypertonic chyme. These inhibitory effects protect the intestine from excessive chyme or acidity. Inhibition is achieved through the enterogastric reflex and enterogostrones, which include hormones like secretin and cholecystokinin (CCK). Neural and hormonal mechanisms regulate the release of gastric juice. The enteric nervous system, sympathetic and vagus nerves, and hormones like gastrin, secretin, and CCK coordinate the production and inhibition of gastric secretions. This complex interplay ensures that digestion proceeds smoothly and efficiently, adapting to the body's needs. Several hormones and paracrines act in digestion, including gastrin, secretin, CCK, somatostatin, and histamine. These chemical messengers regulate various aspects of digestive activity, from enzyme secretion to muscle contraction. Understanding their roles helps explain how the digestive system responds to different foods and conditions. Digestive system relies on a variety of hormones and paracrines to regulate its functions. These include not only gastrin, secretin, and CCK, but also serotonin, motilin, and vasoactive intestinal peptide (VIP). Each plays a unique role in controlling muscle contractions, enzyme release, and blood flow. This hormonal network ensures that digestion is coordinated and responsive. Parietal cells in the stomach pump hydrogen ions, H+, into the lumen via proton pumps, while bicarbonate HCO3 is exported back to the blood. Chloride ions Cl join H+ in the lumen to form hydrochloric acid HCl. The resulting increase in bicarbonate in the blood is known as the alkaline tide. This mechanism is essential for creating the acidic environment needed for protein digestion and defense against pathogen. Stomach responds to filling by stretching to accommodate incoming food. Two factors keep pressure constant until about 1.5 liters of food are ingested: receptive relaxation and gastric accommodation. Peristaltic waves move toward the pylorus, and contractions are most vigorous near this region. Only liquids and small particles pass through the pyloric valve, ensuring controlled delivery to the duodenum. Peristaltic waves move food toward the pylorus at a rate of 3 per minute. The basic electrical rhythm is set by enteric pacemaker cells, ensuring coordinated contractions. Distension and gastrin increase the force of contraction, helping mix and propel food. This activity is crucial for breaking down food and preparing it for absorption in the small intestine. Duodenum controls how much chyme enters, preventing overfilling. Duodenal receptors respond to stretch and chemical signals, activating the enterogastric reflex and enterogostrones to inhibit gastric secretion and filling. The stomach empties in about 4 hours, but fatty chyme can increase this time. Carbohydrate-rich chyme moves quickly through the duodenum, highlighting the importance of food composition in digestion. When your stomach growls in response to the sight or thought of food, the cephalic phase of gastric secretion is initiated. This phase prepares the digestive system for incoming food, demonstrating how external stimuli can trigger internal responses. The cephalic phase is an important example of how the brain and digestive system communicate. Liver, gallbladder, and pancreas are accessory organs associated with the small intestine. The liver produces bile, a fat emulsifier, the gallbladder stores bile, and the pancreas supplies most enzymes needed to digest chyme, as well as bicarbonate to neutralize stomach acid. These organs work together to ensure efficient digestion and absorption of nutrients. Hepatocytes in the liver produce about 900 ml of bile per day, process blood-borne nutrients, store fat-soluble vitamins, and perform detoxification. For example, the liver converts ammonia to urea, stores glucose as glycogen, and makes plasma proteins. These functions are essential for maintaining metabolic balance and supporting digestion. Bile is a yellow-green alkaline solution containing bile salts, bilirubin, cholesterol, triglycerides, phospholipids, and electrolytes. Bile salts are cholesterol derivatives that function in fat emulsification and absorption. Bilirubin, a pigment formed from heme, is broken down by bacteria in the intestine to stercobilin, giving feces their brown color. The enterohepatic circulation recycles bile salts, conserving resources and ensuring efficient digestion. Enterohepatic circulation is a recycling mechanism that conserves bile salts. Bile salts are reabsorbed into the blood by the ileum, returned to the liver via hepatic portal blood, and resecreted in newly formed bile. About 95% of secreted bile salts are recycled, with only 5% newly synthesized each time. This efficient system supports ongoing fat digestion and absorption. Pancreas has both exocrine and endocrine functions. Exocrine cells produce pancreatic juice, which contains digestive enzymes and bicarbonate to neutralize stomach acid. Acini are clusters of secretory cells that produce zymogen granules with proenzymes. Duct cells secrete water and bicarbonate. Endocrine cells found in pancreatic islets secrete insulin and glucagon to regulate blood sugar. This dual function makes the pancreas essential for digestion and metabolism. Pancreatic juice is produced in amounts of 1,200–1,500 ml per day. It is a watery alkaline solution pH 8 that neutralizes acidic chyme from the stomach. The juice contains electrolytes, digestive enzymes for proteins, proteases, carbohydrates, amylase, lipids, lipases, and nucleic acids, nucleases. Proteases are secreted in inactive forms to prevent self-digestion and are activated in the duodenum. This ensures safe and efficient digestion of all food types.